All right, everybody, here we go with non-Mendelian genetics, part 1. So up until now, we've been talking about the laws of Mendel and how everything is this way and that way and dominant and recessive. And those conditions apply to a lot of circumstances, a lot of genetic inheritance, but not all. So we're going to look at some exceptions to those rules. Okay, so Mendel said each trait is completely controlled by a single gene. Only 2 possible alleles exist, and one allele is completely dominant over the other allele, which is recessive. Okay, so this is true for a lot of stuff, but not always. So again, most traits are influenced in more varied and subtle ways, and almost all human genetics is non-Mendelian genetics, kind of different rules apply. So first we're going to get into incomplete dominance. And this is basically where there's not a complete masking of the recessive allele. So the phenotype of a heterozygote is in between the phenotypes of the homozygotes. Okay? So Mendel said one allele was dominant over the other one was recessive. But there are a lot of alleles that are incompletely dominant. They can't completely mask the recessive allele. So if the heterozygous phenotype is intermediate or between the 2 homozygous phenotypes, so say I have a red flower and a white flower, and if I have a homozygote, one red allele, one white allele, my flower would be pink. So it would be a blending or an in-between phenotype. Okay, so that's incomplete dominance. So we're going to look at human hair texture. Has 2 incompletely dominant alleles, H1 and H2. Okay, H1, if you're homozygous for H1, you have curly hair. If you are homozygous for H2, you have straight hair. If you are heterozygous, 1 curly allele and 1 straight allele, you have a wavy hair. So it's kind of in between. So if 2 wavy hair people married, that's heterozygotes, so it's a monohybrid cross. Their children, let's look at this. So we have wavy hair mom, heterozygous, wavy hair dad, heterozygous. Okay, so their children, let me creep this up a little bit, can have curly hair, so homozygous for curly, straight hair, homozygous for straight, and then the heterozygotes will be wavy. Okay, so it's an in-between phenotype. It's kind of a blending of the 2 other phenotypes. Okay, so that is incomplete dominance. Another example of non-Mendelian rules is that a single gene can actually have more than 2 alleles. Okay, you, an individual in your genome, can only have 2 different alleles or 2 the same. Okay, but you can't have more than 2 alleles of a given gene because you only have 2 homologous chromosomes. They come in pairs. But a species, okay, can have multiple alleles in the gene pool. Okay, and if you think about things in humans like hair color, eye color, there's more than 2 colors of eyes. There's more than 2 colors of hair. So there has to be more than 1 allele. Okay. So we're going to look at blood type as an example of a gene with multiple alleles. And there are actually 3 alleles for blood type. Okay. So there are 3 alleles for the system, the A allele, the B allele, and the O allele. And these 3 alleles can produce 4 different blood types. Blood type A, blood type B, blood type AB, and blood type O. So let's see how this works. There's actually another rule that we're going to talk about that comes into play here. But first, we're going to look at multiple alleles. So 2 alleles, A and B, code for enzymes that add sugar molecules on the end of glycoproteins that protrude from the membranes of red blood cells. You guys remember glycoproteins from back in our plasma membrane chapter? Okay. So that's what A and B for. They code for enzymes that add sugar molecules, different sugar molecules, to glycoproteins. Okay. The O allele codes for a non-functional enzyme that doesn't add a sugar molecule. Okay, so the blood types arise as a result of what type of sugar molecules you have on the glycoproteins on your cells. Okay, so allele A and allele B are both completely dominant to O. Okay, so A is dominant to O and B is dominant to O. So if you have the genotype homozygous A or heterozygous AO, you have blood type A. If you have homozygous B or homozygous B or heterozygous BO, you have blood type B. Okay. The only way you can get blood type O, because it's recessive, is to be homozygous recessive. So that's how you get blood type O. Okay. Now, if you have, so we've covered almost every combination except combining the A and the B allele together, that heterozygous combination. If you have one of each of the A and the B allele, you actually have AB blood, okay? And that's because the A and B alleles are codominant to each other. So what codominance means is that if you have codominant alleles, an A allele and a B allele, the individual is going to exhibit the phenotype of both genes. So it's not a blending like incomplete dominance. It's the distinct phenotype of both genes. Okay. We'll talk about another example of this in class. Okay. So because of that, the plasma membranes have both A and B glycoproteins. Okay. And again, we call this codominance. So how blood types work, and we're going to do a little bit of a blood type worksheet in class, is that people can make antibodies to the glycoproteins that they don't have. Okay, so you've heard about like different people can give blood to different other types and you can only get certain type blood. And that has to do with the antibodies. So if I have type A blood, which I do, I make antibodies to the B glycoproteins. So basically, if you put type B blood inside me, my immune system would attack it. And it's never good when your immune system attacks your own blood. You kind of die. Okay. People with type B blood make A antibodies. Okay. People with O blood make both A and B antibodies. Okay. And if you have AB blood, you have both glycoproteins, you make no antibodies. Okay. And what this does is this kind of gives me the laws of who can donate blood to whom. Okay. And again, how the antibodies work, they cause the red blood cells to clump together, rupture. Again, this is never good in your blood. And actually, you're not even sad, you're just dead. Blech. Okay, it's no good. So you have to be very, very careful about getting blood. Okay. So this is, again, you know what, I think I'm just going to go to the next slide because I think it can explain much better. Okay. But yeah, I'm just going to go to the next slide. So you can always come back or pause here and read this up, but I can explain it better with this. So here, if my blood type is A, these are my different genotypes, and I have A glycoproteins, and so I make B antibodies. So I can receive blood from A or from O because they don't have any B glycoproteins. Okay. I can donate to somebody who is A or AB. Blood type B, genotypes, again, has antibodies to A. So we can receive blood from B or O and donate to B or AB. AB, okay, has both A and B glycoproteins, and they have no antibodies, so they can receive blood from everybody, and they're called the universal recipient. Okay, they can only, however, donate blood to other people with AB. Blood type O has no glycoproteins. It makes both A and B glycoproteins. So it can only receive blood from O, but it can donate to everybody. So they're called the universal donor. Okay, so you hear sometimes in the hospital dramas, hang a bag of O-neg, and so that's if you don't know somebody's blood type, you can always give them blood type O. Okay, so we'll do a nice handy chart when we're going to go over this example in class. But so the blood group is an example of multiple alleles and codominance. Okay, and then there's also the example of incomplete dominance. Okay, and so that is end part 1. Go get them with part 2.